Assalamualaikum and hello everyone. I am Madam Suraya Iliani binti Muhammad Suhud. Let's learn about mutation. There are 3 subtopics in mutation, and in this video, I will cover 2 subtopics which are 7.1 and 7.2. We start with subtopic 7.1 first. Subtopic 7.1 is about mutation classification and types. The learning outcomes are: define mutation, classify mutation to gene or point mutation and chromosomal mutation, state 2 types of mutation, spontaneous mutation and induced mutation, define mutagen, state types of mutagen. Mutation is a spontaneous change in the amount, arrangement or structure of the genes or DNA of an organism. DNA carry genetic information for protein synthesis. If the DNA is mutated, this information will interrupted, resulting in formation of abnormal protein or no protein produced. When this happens, it may lead to change in genotype and phenotype. Mutation can be classified into 2 main classes, which are gene or point mutation and chromosomal mutation. Gene or point mutation has 4 classes. The first class, known as base substitution, next is base insertion, base deletion, and lastly is base inversion. Chromosomal mutation has 2 main classes. The first class is chromosome aberration, and the second class is alteration of chromosome number. Chromosome aberration can be classified into translocation, deletion, inversion, and duplication. Alteration of chromosome number has 2 main classes. The first class is aneuploidy and the second class is euploidy, or also known as polyploidy. Euploidy can be further classified into autopolyploidy and allopolyploidy. There are 2 types of mutation. The first type is spontaneous mutation and the second type is induced mutation. Spontaneous mutation is a natural changes in DNA structure and occur at a lower rate, while induced mutation is a damage of DNA that lead to alteration of gene caused by mutagens. Induced mutation occur at higher rate. Spontaneous mutation can be occurred due to error during DNA replication when the nucleotide base pair up with non-complementary bases and the errors are not corrected by DNA repair system. It also can be occurred due to non-disjunction during meiosis. Non-disjunction occur when homologous chromosome or sister chromatid fail to separate during meiosis 1 or meiosis 2. Induced mutation occur due to mutagen. Mutagen is a chemical or physical agent that causes mutation, thus increases the rate of mutation. There are 2 types of mutagen. The first one is physical mutagen and the second one is chemical mutagen. The example of physical mutagen are UV ray, gamma rays, and X-ray, while the example of chemical mutagen are colchicine and ethidium bromide. Now, let's proceed to the subtopic 7.2, which is gene mutation. Subtopic 7.2 is about gene mutation. The learning outcomes are: define gene mutation, state the 4 types of gene mutation, and state the effect of base substitution, which lead to missense, nonsense, and silent mutation, and the effect of base insertion and base deletion, which lead to the frameshift mutation. Gene mutation is a change in a single nucleotide pair of a gene, lead to the change in amino acid sequences. For example, for normal hemoglobin, one of the base triplet is CTC. During transcription, this base triplet will be transcribed to codon GAG. GAG encodes for amino acid glutamine. Therefore, the shape of red blood cell is normal, which is biconcave shape. But for the sickle cell hemoglobin, mutation occurs when thymine is replaced by adenine. Therefore, the base triplet is CAC. During transcription, this base triplet will be transcribed to codon GUG that encode for amino acid valine. Therefore, the structure or the shape of the red blood cell is abnormal, which is sickle shape. Gene mutation is classified into base substitution, base insertion, base deletion, and base inversion. Base substitution is occur when a pair of nucleotide is replaced with another pair of nucleotide. For example, a pair of nucleotide that consists of cytosine and guanine replace the fourth base pair in normal DNA. Next is base insertion. Base insertion is addition of one or a few nucleotides in DNA sequence. Cytosine and guanine is inserted between the fourth and the fifth base pair. Base deletion is loss or removal of one or a few nucleotides in DNA sequence. The example is the fifth base pair, which is thymine and adenine, is deleted from the normal sequence of DNA. Lastly is base inversion. Base inversion occurs when two or more nucleotides are inverted, which is rotate 180 degrees. The example is the third and the fourth base pair, which is cytosine, guanine, and thymine and adenine, are rotated 180 degrees. So as the result, the position of base pair are exchanged. Now, let's see the effects of base substitution. When base substitution occurs, it may lead to another type of mutation, either missense mutation, nonsense mutation, or silent mutation. Missense mutation is a nucleotide pair substitution that results in a codon that codes for a different amino acid. For example, cytosine is replaced by adenine. During transcription, the codon produced is a codon that codes for amino acid threonine instead of proline. Nonsense mutation is a mutation that changes an amino acid codon to one of the three stop codons, resulting in a shorter and usually non-functional protein. For example, cytosine is replaced by guanine in DNA, and during transcription, the codon produced is a stop codon. Stop codon is a codon that not encode for any amino acid. For silent mutation is a nucleotide pair substitution that has no observable effect on the phenotype, and within a gene, a mutation that results in a codon that codes for the same amino acid. For example, adenine is replaced by thymine, and during transcription, the codon produced still code for the same amino acid, which is valine. Now we look at the effect of base insertion and base deletion. These two types of mutation can lead to frameshift mutation. Frameshift mutation is a mutation occurring when nucleotides are inserted in or deleted from a gene, and the number inserted or deleted is not a multiple of 3, resulting in the improper grouping of the subsequent nucleotides into codons. The example is, this is the normal sequence of DNA, and during protein synthesis, the sequence of amino acid produced as shown in this figure. When base deletion occurs, guanine is removed from the second base triplet of DNA and resulting in the improper grouping of the subsequent nucleotides into codon. So, the sequence of amino acid change entirely start from the point of mutation. Same goes to base insertion. When Adenine is added to the second base triplet of DNA, so the sequence of amino acid produced is different from the normal sequence.
Now we at the end of this video. Hopefully all the learning outcomes are achieved. To test your understanding about this lesson, you may try these two questions. Thank you.